Ribosomes are great. They're made of a mix of RNA and protein. Stick a 30S subunit and a 50S subunit right here on this Shine-Dalgarno sequence, or SD sequence, which is where translation begins, and watch it go. Wow, free proteins. Ribosomes are great at mass-producing proteins for us, like insulin or antibodies. People have engineered genes to do better things for us, so what if we could also engineer ribosomes? What if we started attaching things to them or changing key residues to make them better? What if we could mass-produce proteins with synthetic amino acids or expand the genetic code? What if we could mutate every single amino acid in a ribosome to better understand ribosomopathies? There's one problem. Changing basically anything about a ribosome might instantly kill you. Ribosome sequences have been conserved across billions of years for good reason. Well, what if we had some ribosomes assigned to do their regular ribosomal jobs, and other ribosomes for us to mess around with and observe? In other words, can we make an orthogonal ribosome? A ribosome that is not responsible for the normal bacterial proteome, and that translates its own special set of RNAs. In 2005, some people did that. They changed the SD sequence that ribosomes recognize at the beginning of translation. Normal ribosomes recognize the classic sequence, GGAGG, through complementary base pairing with a region at the 3' end of the 16S rRNA, known as the anti-Shine-Dalgarno sequence, or ASD. Changing the ASD to something else resulted in the ribosome not binding regular mRNAs at all, and only binding to those with a complementary SD. In other words, this was an orthogonal translation system. So, that's great. You have an orthogonal 30S subunit, but these can still interchange with wild-type 50S subunits. In other words, we could screw up 30S subunits as much as we wanted, but screwing with 50S subunits could still affect cellular health. So it wasn't totally orthogonal just yet. In 2015, some people thought, well, why don't we just tie them together? They literally created a tether between the orthogonal 30S subunit and its 50S subunit by fusing together the 16S rRNA to the 23S rRNA. It looked like this, and it worked. No matter how hard you try, you can't stick a wild-type subunit on it. This ribosome, named RiboT, was the first truly orthogonal artificial ribosome. So, you might ask, isn't it kind of weird if you tie these two together? There must be a reason why they stayed separate all these years, right? Well, you would be right, because RiboT was actually garbage at being a ribosome. Bacteria with RiboT could survive, but it only translated proteins at half the rate. It's because it became more slow at departing from the start codon. This meant that trying to edit these ribosomes and observe the results wouldn't give us an accurate scientific picture at all. So, to fix this, the same team wondered, what if we used both kinds of ribosomes that have been designed? If we can't stop the orthogonal 30S subunits from exchanging with normal 50S subunits, then let's just have RiboT recognize the wild-type mRNAs and make the dissociable subunits the orthogonal system. Thus, the OSIRIS cell line was born, which stands for Orthogonal Translation System based on ribosomes with isolated subunits. RiboT kept the cell alive, and the mutant 30S subunit, which they called the O ribosome, was the orthogonal system. The O ribosome had one mutation, which was A2058. Mutants with just this mutation and no RiboT had no transformants, but those with RiboT did appear, showing that A2058 was definitely lethal. They inserted GFP reporter genes with an orthogonal SD and found that their expression relied on the presence of the O ribosomes, with a little bit of recognition by RiboT, but it was a drastic difference compared to expression in wild-type cells. Furthermore, the O ribosome was an improvement upon orthogonal RiboT cells. Even when O ribo was in a low copy number plasmid, meaning that less of it was transcribed, it produced more OGFP than O ribot in a high copy plasmid. To definitely prove the orthogonality of OSIRIS, they tried to murder their cells by adding known lethal mutations into the 50S subunit that knocked out the peptidyl transfer site, and the cells survived fine on ribot. Finally, they used OSIRIS to try to find gain-of-function mutations. More specifically, they wanted to engineer a ribosome that would more easily release proteins that are difficult to terminate, which we might need if we want to translate synthetic amino acids. An example of this is during translation of TNAC, which is a regulatory tryptophan synthesis protein. At excess concentrations of tryptophan, TNAC release is inhibited, and the ribosome stalls at the stop codon. This leads to activation of the TNA operon, which catabolizes tryptophan instead of synthesizing it. And so they constructed a reporter where TNAC was placed at the end of a GFP gene, so that the GFP fusion was less expressed at high tryptophan levels. They then made a library of 120 point mutations in O ribo's peptidyl transfer site. and of those, 19 were found to have better translation of the GFP fusion than wild type. So ultimately, what do you think we can do with orthogonal systems like OSIRIS? It might be a new era of possibilities, from understanding the ribosomal errors that cause disease, to learning the basic science behind translation, or creating whole new categories of proteins never seen before.